All right, why don't we go ahead and get started? So thanks everyone for taking the time to join. We've got a couple people still coming in. I imagine everybody here is either a clinical research professional or would like to be a clinical research professional and has at least a passing interest in genetics and genomics. So what we're going to do today is I'm going to run through at a pretty high level some of the basic concepts to start with, and then we'll dive into some of the most important technologies and I think areas that I'm excited about in the future when it comes to the intersection between genetics and clinical research. So a little bit about me and my background. My PhD is in population genomics. So I'm American, as you may be able to tell from my accent, but I moved over to the UK about 10 years ago and worked on a number of large scale population genome programs focused on rare disease, common disease, and everything in between. And this was based out of the Sanger Institute and the University of Cambridge, which is the Sanger Institute is a big sequencing center just outside of Cambridge in the UK. And I co-founded Sano in 2017. And what we do in a nutshell is help clinical research teams to accelerate their precision medicine trials by combining genetic testing, recruitment, and long-term engagement into one platform. So I'm not going to spend very much time. I think just one slide on our platform so you know a little bit about me and what we do. But the most of today is going to be about genetics and genomics more broadly in clinical research. And if you do want to learn more about us, then you can email me. I'll have my email at the end, or you can reach out through our website. So I want to start with a personal story. So this is an article from the New York Times in 1984. And one of the families mentioned in this article is my family. My grandfather specifically and his brother were amongst the first kidney transplants that were conducted in New York. And at the time, my grandfather was he was the head of security at Memorial Sloan Kettering, which is a famous medical center, famous cancer center. And I think at the time they sort of thought nothing of it, but over the generations, they started to notice that many members of the Short family ended up with kidney transplants. Almost everybody who was either receiving a kidney or donating a kidney if it didn't seem like they needed one. And so this was a little bit of a personal introduction for me into genetics from a very young age. I was always hearing about our kind of mysterious rare kidney disease that ran in the family because we had been in research projects. It wasn't any of the obvious genetic kidney diseases. And I'm not going to spend a lot of time on this, but actually after enrolling in a genetics research study now nearly 7 years ago, we very recently got an answer to the genetic driver behind our family's kidney disease. But the reason I bring this story up is because I've worked as a researcher for a long time, but I also had a front row seat to what it's like to be a participant in a series of genetics and precision medicine studies, both both small and large. And as a participant, one of the things I saw firsthand is how much opportunity there is to innovate, not just on the research side where I've spent most of my career, but also some of the basics and fundamentals of actually what is it to be a recipient of a genetic test, to get results back, and in the context of something that's as fast changing as clinical research. So in 2017, along with my co-founders, I started Sano, and what we do is we help companies that are running natural history studies, registries, clinical trials that are involving precision medicines to do recruitment, genetic testing, and patient engagement. So I'm not going to spend much time on this, but just to orient you about us and what we do if you're new to Sano and what we do. Okay, so focus for today. I'm going to go through really five, and then Q&A at the end, but 6 main sections. So I'm going to talk about some of the trends and drivers behind why genetics and drug development is becoming such an important topic. So for those of you in the room, I suspect you're either working in clinical research or thinking about it, and may already today be working on a clinical trial involving a precision medicine. But if not, this will hopefully give you a picture of what's coming. And then we're going to go through core genetics concepts, some of the relevant technologies, and then finish off with applications across discovery, development, and delivery, by which I just mean moving into the healthcare system. So before I jump in too far, you all have probably used Zoom before. Feel free to put questions in the Q&A or in the chat, and I'll stop a couple times throughout to answer questions if there are that come through that are relevant to the particular section. Otherwise, we're going to have hopefully plenty of time at the end to answer questions as well. So some of the trends, you may have seen graphs like this before, but we've seen an enormous shift in the last 13 years. So in 2005, just 5% of new therapies that were approved by the FDA would be considered personalized using biomarkers, genetics being one of the main ones to personalize the therapy. And now, and fast forward to 2018 and in the 2020s, we're seeing the number of precision medicines really skyrocket. And I think in the next couple of years, it's likely that we're going to tip the balance into greater than 50%. And then ultimately, I think in the next decade, we'll probably live in a world where the majority and the vast majority of medicines come with some kind of genomic biomarker or other biomarker that really hones in on a particular population that's more likely to benefit from the treatment. So if you're not working on a precision medicine today, it's very likely that you will be in the next 5 years if you do work in this field. And one of the things that's driving this is the cost of sequencing a human genome is has dropped exponentially. So at the start of this graph is the early 2000s, and the Human Genome Project was completed at that point, and it cost at the completion of the Human Genome Project more than $100 million per genome to sequence a single person. And the Human Genome Project itself actually cost more than $1 billion. But what you can see on this graph is something very dramatic happen. So Moore's law is the reduction in cost and the increase in efficiency of computing power over time. It's something we've all seen firsthand, as the computer that helps with the moon landing is roughly the same size as the computer that's in our phone. I think the phone's probably way stronger now. And the same thing has happened with genomics, but actually on an even more extreme scale. So in the space of 20 years, we've gone from $100 million to sequence a single person, now down to less than 1,000. And for those of you who work in clinical research, I think you can understand and appreciate the implications that this has. If it even costs $10 million to sequence a single patient, then this would make it literally impossible to run any modern rare disease or precision medicine studies that involved exome sequencing or whole genome sequencing. And even if it had stuck on Moore's law, it would be a different story if it had been $100,000. So the exponential reduction in costs and the improvement in technologies has really been a big part in driving this wave. And off the back of this, one of the things that we've learned as a field, there have been a number of large pharma that have looked retrospectively at their portfolio to try to get an understanding of the impact that genetics has on success in clinical trials. And they found across the board that genetically validated targets, meaning there's a strong understanding of the genetics that drives disease, results in 2 to 3-fold higher success rate in clinical trials. And this is due to a couple reasons. The first is that these kinds of targets tend to actually be safer, because if you have genetic evidence that shows that you could knock this out in humans and they're healthy, then that gives you support that you may be able to knock a particular gene or protein down with a small molecule, and it's also likely to be safe. And we're going to go into an example of this in a little bit more detail later. Another reason is greater predictability of efficacy, that the target is actually linked to disease, and that modulating that gene or modulating that protein is actually going to have an impact. So what the genetics provides in the early R&D stage here is a really clear link between gene or protein and the disease in question, and often the mechanism in question as well. And then there's another piece around protocol design. So rather than running one drug for an entire population that's undifferentiated, there have been a lot of great innovations, in particular in oncology, in protocol designs that take a broad population of patients, stratify them based on one biomarker or a combination of biomarkers, and then actually place them into the right treatment arm for a clinical trial. So all of these things together have contributed to genetics becoming a big part of the discovery and development engine in both large pharma and biotech. And I want to give a couple of concrete examples here. So there's a very famous one dating back to the early 2000s, which is a gene called PCSK9. And what was discovered here essentially is that this gene was altered in families that have hypercholesterolemia, so high cholesterol. And it was also discovered that people that have this gene knocked out or knocked down would actually have low cholesterol. And you can see the logic here, that if in some families the gene causes high cholesterol, and in some knocking the gene out causes lower cholesterol, then it leads to a very strong hypothesis that inhibiting PCSK9 with something like a small molecule could actually have a causal relationship to reducing cholesterol. And what's really important here is I want to deconvolute the concept of gene editing and some of the modern therapies with precision medicines, because in this case, this is a small molecule in and this was happening in the early 2000s. So genetics here is being used to really understand the biology, and we will talk a little bit more about modern therapies that go in and edit genes directly. There are a couple other examples here. I won't spend too much time on the second one. It's similar. A genome-wide association study was used to identify a gene that was involved in not just Crohn's disease, but it turned out a number of other autoimmune diseases. And this discovery led to a number of monoclonal antibodies that are being used still today. And then this third example is one of my favorites because it's an example of the human knockout study that I mentioned earlier. So there's a rare disease called PH1, and in order to treat this disease, the researchers working on this believed that they needed to knock down this gene called HAO1, but they had questions about whether knocking the gene down was healthy or what was going to contribute to off-target effects or side effects. And what they discovered was in a biobank in London called East London Genes and Health, they discovered an individual who had both copies of this gene knocked out. So they had loss of function mutations. It just naturally, it's almost like a natural experiment that happens by randomness. And by recalling this participant, collecting new samples, and also looking at health records, they were able to determine that this individual did not have any dramatic comorbidities as a result of having this gene knocked out their entire life. And so this is a really useful piece of information as part of a portfolio of toxicology data to say, okay, if somebody can lack this gene entirely and be okay, then it probably stands to reason that we could selectively knock this gene down in a sick population, and we're less likely to have severe off-target effects. So there are dozens of examples like this. I wanted to highlight three that I think are kind of representative of some of the opportunities of genetics in development. Okay, so moving into some of the core genetics concepts. For some of you, this may be new. For many people, this may be a refresher, but I'm going to start relatively simple, but we won't be here for too long. So if you already know all this stuff, then please bear with me. So children receive 2 copies of every gene, with the exception of the sex chromosomes. But if you take almost every gene in the genome, we get 1 copy from mom and 1 copy from dad. So you can see this family tree on the right-hand side here. And I'm not going to go through exactly how to read these, but in this case, you're seeing an example of a simple trait or Mendelian disease that's passed down, and the coloration of the tree on the right shows here whether the person has or doesn't have the gene in question. And you'll be familiar, I think, with many examples of monogenic disease. A lot of them are rare, things like cystic fibrosis, sickle cell, Duchenne muscular dystrophy. More and more monogenic forms of common disease are being discovered as well. So monogenic forms of obesity, where there's a single gene that is, when it's mutated or changed in the right way, has the ability to induce what would maybe be considered a more complex or common disease like obesity. And there are also classical examples like hypercholesterolemia. And there are literally thousands of diseases that fall in this category. Many of those are rare and ultra rare, but depending who you ask, there's at least 6,000 rare monogenic diseases, and the number is growing. There's 2 essential concepts that I think is important if you work in this field to understand when it comes to the more simple genetic diseases, quote unquote. The first is prevalence, and the second is penetrance. And prevalence is a relatively simple one. So I want to use an example here of a disease that has a strong genetic basis, which is FSGS, a form of kidney disease, and the gene is called APOL1. And what you can see on the map here is that the gene variant, the set of variants that cause this disease, is a lot more common in parts of West Africa, and also therefore in parts of the Americas where there's large populations of West African ancestry. And what you can see in contrast across much of Europe and Asia, if you look on the legend on the right here, that genetic variant is very rare, or in some cases totally absent. And so what this drives is, depending on which countries you are or which populations you're looking in a particular country, you're going to get vastly different prevalence rates of this particular genetic disease. And this obviously has implications on how you enroll clinical trials, how you create your recruitment plans, but it also has an impact on some of the fundamental genetics R&D work that you do. If you don't have a representative data set of Americans, for example, then you're likely to miss diseases that are more common in underrepresented groups. And this is far from the exception. Actually, many genetic diseases are at very different prevalences in different parts of the world. And so this can affect all kinds of parts of your clinical research plan dramatically. So it's really important to not just understand the prevalence at a broad population scale in somewhere like the US or Europe or any part of the world, but actually to really understand it on a global level. The second concept is penetrance. This one is a little bit less intuitive maybe than prevalence, but still I think pretty straightforward. So penetrance is about what percentage of people that actually have the genetic variant will go on to get the disease. So if something is fully penetrant, then it means if you have the gene, you're going to end up with the disease. And if something is incompletely penetrant, then it means that having the gene doesn't necessarily mean you go on to get the disease. And as you can imagine, the levels of incompletely penetrant can vary dramatically. So some genes and diseases, you may have 90% penetrance, or to use this example here with the LRRK2, G2019S mutation in Parkinson's disease, by 80 years old, this gene is at about 74% prevalence, 74% penetrance, meaning if you have this gene, then 3 in 4 people who have the gene will develop the disease by the time they're 79 years old. But it's only 28% penetrant by age 59. And there are other diseases that are near 100% penetrant and others that are 20, 30, 40%. And we're starting to blur the lines as a field right now between monogenic and polygenic, which I'm going to touch on what I mean by polygenic in a second. But again, this is a really important concept because we see a lot of teams get tripped up on the difference between having the gene, having the disease. And in many of these cases, there's actually a 2x2 grid that you need to draw where you can have the disease and the gene, you can have the disease but not the gene, but you can also have the gene but not the disease. And it's really important to get these right because it has implications on the patient journey in your studies and your clinical development programs, and also how you build out the plan to actually get your medicine in the hands of patients if it is approved. So I'm going to spend one slide on polygenic risk scores because I think they're an important emerging trend. They're not yet very widespread certainly not in clinical trials. I'm starting to see them a lot more in earlier stage R&D, but the essential concept here is to take hundreds, thousands, or even in some cases, millions of genetic variants across the genome that individually have a small impact on a trait or on a disease. So they may only increase your risk on their own by 1% or a fraction of a percent, but actually you can algorithmically combine these scores together and add them up to get people who happen to carry a large number of these tiny pebbles that add a small amount of risk, but they've inherited hundreds of them or thousands of them that push them higher on the risk scale. So you would never detect this in a typical family tree, or it would be hard to detect it in a typical family tree. It doesn't have the kind of pattern that you'd expect of something that's highly penetrant, where if you have the one gene, you always get the disease. So this is emerging as a pretty important topic area. We have run a number of podcasts on this topic. The one I'd recommend is episode 78. Peter Donnelly is one of the pioneers in this field, and gives a really great overview on what it is and where it's going. Okay, so we talked a little about some of the applications. I'm going to go into some of the technologies now that are most relevant. So first I'm going to start with DNA sequencing, and then I'm going to talk about sample collection and return of results. So 2 and 3 may not seem like technologies in the same sense that DNA sequencing are, but I hope I'll convince you that it's important that they are, because if you're not actually able to collect the samples effectively in a remote environment and return results, then it doesn't really matter how sophisticated your analysis is. I'm not going to go into too much detail around the nuts and bolts of things like gene editing and how to deliver some of these novel therapies into affected tissues. We may do a follow-up on this if people think it's interesting, but we're going to focus on these first few today, as they tend to be the most important part in a lot of transitions from early stage R&D to clinical trials. So I showed this graph earlier. I'm just going to briefly revisit it. Where we are today to do very comprehensive genome sequencing, the actually running the sample on the machine now costs less than $1,000. There's other costs associated with sample collection, counseling, analysis, but I just want to reiterate how far we've come. And there are programs today that are sequencing hundreds of thousands and some approaching, or with the plan to approach millions of participants, that just simply wouldn't be possible if sequencing wasn't this expensive. So I'm going to go through a couple of the common ways that we read DNA and what the differences are. And then I'm going to summarize this in a table at the end. So the simplest, the lowest cost, and the highest volume kind of testing that is typically done is something called genotyping. So if we look at this hypothetical string of DNA here, we have 6 billion letters in our genome. And what genotyping does is it hones in on between 500,000 and a couple of million known locations in the genome, and just tells you what the person has, what letter they have at that location. So at this location, our hypothetical reference sequence, we call it, has a T, and the person we're testing, we know, has a G. And at this location, the hypothetical reference has a C, and the person we're testing knows that we have an A. And so this works across millions of locations in the genome. And there are some clever things you can do to actually fill in the blanks in between these letters using computational methods. I'm not going to spend any time on that today, but what this technology is really good for is for testing at a low cost and high throughput for things that we essentially already know about. So if we happen to know that this part of the genome is involved in a common or rare disease that we're interested in, then with genotyping arrays, we can test for that really quickly and at the lowest cost. So the second important method is something called exome sequencing. So if we start again with this hypothetical reference sequence, in our 6 billion letters of DNA, only about 2% of those actually encode for proteins. So the other 98% does things like controls the amount of protein that's produced, or in some cases, in many cases, actually, it's considered junk and it doesn't do anything at all. But it turns out that a lot of the action in the genome is in about 2% of the 6 billion letters. So what exome sequencing does is it hones in on those sections of the genome and ignores everything else. So if you can imagine, again, to use this simple example, that this is one gene and this is a second gene, and all the stuff in the middle is what we've cut out because it's not coding for a protein. It's not ultimately going to turn into a protein. What exome sequencing does is it goes very deep and it gets every letter within all 20,000 protein coding genes in the genome. So for most of the monogenic diseases that I listed to you earlier, things like cystic fibrosis, Duchenne muscular dystrophy, sickle cell, these are all affecting the protein and would show up in an exome sequencing test. And where these differ from the genotyping that I showed you before is you don't just get a single letter like we get before, you get the whole string. And that allows you very often to pick up rarer changes and things that you didn't know about before. Whereas, as I mentioned, with the genotyping array, you need to have known about it before in most cases in order to detect a change. So that's exome sequencing. And I won't have a slide for this, but there's a version of exome sequencing which doesn't do all 20,000 genes, but just does a couple hundred. And that's typically called panel sequencing or a gene panel. And I'll summarize that in the table later. But in case you hear this term, you can think of it like exome sequencing, except it's a fewer number of genes. And these were very popular in the maybe 5 to 8 years ago, because they were a very cost-effective way of doing this sequencing on a set of a couple hundred genes that you know about. These days, more and more exome sequencing is being done because the approach and the cost is roughly on par with panels most of the time, and you get so much more information. And then the final one here is whole genome sequencing. So this is like the best of both worlds. The genotyping arrays do get information in between the protein coding genes, but it's not deep. The exome sequencing only gets the coding regions, but it goes very deep, and you can pick up rare variants. Whole genome sequencing picks up nearly 100% of the DNA. There's still some parts that are challenging to sequence because they're repetitive and so on. We're not going to spend any time on that today. But again, I'm zooming in on the same part of the genome, and what you'd learn here is different in a whole genome than what you get from an exome. And this is of the three, this is obviously the most expensive, but it's still insanely cost effective compared to where it was just 20 years ago. And I just want to show you one interesting one for fun here. And I think it also maybe hits home how sequencing works. So this is what I'm showing here is a strand of DNA getting passed through. This is called a nanopore, so it's getting passed through a protein that's on that's basically sits on a membrane, and this little funny signal processing thing here is measuring the change in voltage. I think it's the change in voltage across the membrane as the DNA strand passes through, and this basically allows us to read the sequence. So this is one of the most advanced forms of technology that isn't really used in clinical care, except in a couple of niche use cases, but it's gaining a lot of momentum. And the main reason it's not being used is because it's still fairly expensive, and it's not as easy to use as some of the short read sequencing technologies, which is what I spoke through before. But I think this is hopefully useful to show you how this process works, and what you get at the end out of a long read sequencing is literally a string of tens of thousands of letters in a row that then allow you to come back and assemble the genome. So just to summarize these 5 methods that I mentioned. So from top to bottom, we get generally more comprehensive and more expensive. And the main applications towards the top here are looking at known variants and often large common disease studies that are testing 500,000 or 1 million people will use this kind of approach because it's the most cost effective to get to that huge sample size. But more and more, we're seeing large programs start to use whole genome sequencing. So the Genomic Medicine Service in the UK is now rolling out whole genome sequencing for a wide range of rare diseases and cancers, which I think is really smart because they're both providing a very comprehensive diagnostic service while also building an amazing research resource. And we tend to see most of what we do in using either exome sequencing or genotyping, or actually combining two. We'll combine the two of them in an approach that we call Exome Plus. So one slide here, or I think maybe 2 slides on sample collection. So a lot of people assume that only blood can be used or only saliva can be used for DNA testing, but actually both are essentially identical. There's some very minor differences. You get more bacteria in saliva, and you can sometimes pick up what people have eaten, but that gets filtered out by modern bioinformatics pipelines. So sample collection can be done in healthcare and clinical sites, and basically leverage all the sample collection that's already being done in a natural history study or clinical trial. But also saliva testing is very frequently done. And one of the advantages of this is that it's done non-invasively with a home kit. So it's important to consider both of those, I think, if you're embarking on an interventional or non-interventional study. And the final bullet point here is about biobanking. So this is becoming a lot more routine. And one of the things that you may know about DNA and genetic data is that it doesn't change over time, which is very unique in the biomarker world, because most things you actually, if you measured it today and you measured it next week, it might change. Or certainly if you measured it today and you measured it in 5 years, if it was any blood-based biomarker, you'd expect it to change possibly dramatically. But our DNA doesn't change, and that means that you can collect a sample now and bank it, and then when the technology changes, you can reanalyze it, and it's just as valid and it's just as useful as the sample that was collected today, which is very unique, and so worth considering and thinking about this, and not just pattern matching on the other biomarkers that you may be used to, where you'd need to collect it every year, for example, or need to collect a fresh sample for it to be relevant. So a couple slides on return of results. This probably seems like a simple one, but for clinical research programs, it's really really important to have clear return of results that are not just clear to the healthcare providers or site staff, but also to patients and participants. So we do a lot of work to try to make these tests clear and understandable. But it's worth thinking about this process, where to fold in pre and post-test genetic counseling. In particular, the reason why this is so important is there are many diseases that are going through a transition into becoming precision medicines where genetic testing is not common in the healthcare system. And so in many cases, there'll be healthcare providers that are, if we take Parkinson's disease as an example, that are not used to using or interpreting genetic test results in their standard practice, because to date, they haven't really been used to to determine what therapy or approach you may take with a patient. And so as this changes, it's really important to focus on that clarity of communication and not just assume that a genetic test once completed will be accurately communicated. And another important point here is around communication, return of results. There's a lot of stakeholders that are involved in any kind of genetic testing process. So you've got the patient, the participant, often their families. This is a critical family discussion in many cases of whether somebody chooses to undergo genetic testing, healthcare providers, genetic counselors, the clinical trial sites if involved in the particular study, and then obviously the sponsor of the study who may be getting aggregated, anonymized results back. So if you don't think through the process from the beginning with all these stakeholders in mind, then you can end up with a process that's not particularly optimized and ultimately impacts enrollment. And one of the things that genetic testing allows you to do is to think a lot more personalized about what kind of research journeys and clinical research that we do build. So rather than a one-size-fits-all kind of approach, you can think about how you layer information about a patient's or a participant's genetics, their diagnosis, and also other information about whether or not they live near a clinical trial site or whether they don't, to build journeys that are a lot more personalized. And going back to my story about our family kidney disease, one of the things I noticed very early on taking part in research was how actually little information as a participant I did get back. Often there were, years would go by without hearing anything. And I know as a researcher that the researchers are working on it, but as a participant who isn't also working as a researcher, you don't always know whether your sample's been lost, whether something's going on, and there's going to be a change in the next couple of months. So it seems simple, but thinking about how to create a personalized experience goes a long way towards retention and enrollment. Okay, so I want to go through some of the applications in discovery, development, and delivery. So these three big buckets. And I'm just go ahead and pop any questions in the chat if you have any. I've just got two more sections here, and then we'll have time for any discussion. So why these three buckets? Discovery is all about how the potential new therapeutic gets discovered in the first place, and includes things like target identification, target selection, all of the amazing chemistry that goes into actually modulating that target. And then moving into development, how do we actually move from a preclinical phase where we validated that we believe this drug is going to have an impact on patients to actually how do we get this through phase 1, 2, 3, or if it's a rare disease, you may just have a pivotal trial and all the supporting evidence that's required there. And then delivery is about the healthcare system. How do we actually make sure that a drug that passes clinical trials and proves to be safe and effective actually makes it into the hands of patients and healthcare providers in a timely fashion? So some of the areas that I think are important and emerging areas to focus on in the field in discovery is using genetics to actually inform target and tissue selection. So I reference AstraZeneca's framework that they call the 5 Rs. And if you Google that, you should be able to find the paper as an example earlier in the presentation. But we're seeing both large pharma and biotechs take a really proactive approach towards using genetic data in their target and tissue selection strategy, because there have been numerous studies now that have found this to be hugely impactful on increasing hit rate through clinical trials. A second area is an obvious one that you hear about all the time. There's a new class of therapies, RNAi, gene editing, that are on the rise, and these are having transformative impacts. I think one thing that is also important to consider here, though, is still the vast majority of quote unquote precision medicines that I see are actually still small molecules and biologics. So it's important not to conflate the two and say precision medicine equals gene editing. Actually, precision medicine is about selecting the right patient population, and by far the workhorse that's still, I think, going to have the most impact, at least for a couple of years while the new class of therapies mature, are the tried and true tools of the industry. And it's really taking a well-known approach, like creating a small molecule, combined with a much deeper understanding of biology to make that change. So something to think about there, not to confuse the two of precision medicine equals gene editing. And then finally, I'm really interested in this theme of using human knockout or natural human experiments, things that we can discover in large biobanks to bolster traditional toxicology studies. I think this is a really very clear use case and something that trips up a lot of drugs in clinical trials that you may be able to detect upstream using genetic data. So in development, as we start to think about clinical trials, natural history studies, even supporting registries, it's important to consider things like genetic screening programs or pre-screening protocols. These can be part of trial protocols. So if you take a case like Parkinson's disease, where we've done a number of programs in the past, the genetic testing is not routine in the healthcare system. So thinking about ways to increase access to genetic testing, this can be part of a trial or can be a totally independent patient finding program or genetic testing program that sits across multiple phases. And this is something that we do a lot of. So if this is something you're interested in, then feel free to reach out to me afterwards. The other major paradigm shift, I think, to focus on is basket and umbrella protocols. So these are protocol designs that take a little bit more time to get set up. They're more complex. They'll often have a number of different sub-studies focused on different biomarkers sitting underneath them. But they have the massive advantage, which is if you're set up correctly, you can drive much more efficient spend on recruitment, spend on site selection, and then also for the patient, it's a far better journey to have a trial arm selected for you based on a biomarker combination rather than having to go through the potentially frustrating experience of screening out of multiple trials in a row because you may not carry the right biomarker. And then the other really interesting trend that I've seen more and more in the last couple of years in particular is companies that are accelerating their development timeline by being smart about how they use natural history study data or biomarkers. So we will have a podcast episode that's coming out about this pretty soon, but what the FDA has gotten a lot, they've been innovating pretty significantly on how they approach, especially rare diseases or rare genetic subtypes of common disease with unmet medical need. And you can actually use great natural history study data to create essentially a synthetic control arm. And this is the kind of thing that you obviously need to speak to the FDA about beforehand. But essentially, if you're operating in a rare disease population, it can create an ethical dilemma to enroll patients on a placebo. And it can create a recruitment challenge that you may not want to have. You may need to recruit literally every patient on the planet to run a properly placebo-controlled trial. And so in this case, using robust natural history data to show what's likely to happen to patients in the absence of any treatment can essentially serve as a supporting evidence to either reduce or in some cases eliminate the control arm entirely. And this can be coupled with other trial designs like open label extensions and so on. But this is, I think it's a super interesting area and important one to watch, which does require a lot of advanced planning to get that natural history study data up and running, or partner with a patient group who's already got it. Then on the delivery side, three other bullet points and things to think about. The strategy of whether to go for a companion diagnostic, where you actually take the diagnostic through clinical trials along with the novel therapy, versus using off-the-shelf testing, LDTs, or the kind of testing that's already happening in some healthcare systems, like in the UK. And this is going to be highly dependent on each program and each disease and what the landscape looks like. But there's a lot to think about, I think, here, and importantly to start thinking about early on rather than waiting until it's too late to understand your diagnostic strategy. Related to this is accessibility of diagnostic tests. So it does no good to get a drug approved and then find that actually patients aren't being tested in the healthcare system and there's no way to find them. And there's also many groups that I think that are learning this lesson in a challenging way and having to go through patient finding programs where they're working to uncover large underdiagnosed populations. So again, this is something to think about earlier on in development as you can, this can help to boost trial recruitment as well, alongside actually ensuring that you've got a patient healthcare population that's ready. And then finally, education awareness. Some genetic diseases, genetic testing is common, even to the point where it's included in heel prick tests and other standard of care. But for the vast majority of genetic diseases, patients need to go and families need to go on a what we call diagnostic odyssey that can take years in many cases to get a genetic diagnosis, either because the symptoms are heterogeneous and it's unclear, or healthcare providers are unaware of genetic testing, or there's barriers in place like access through insurance and so on. And a lot of these can be solved through things like pre-screening programs that we mentioned earlier. So how am I doing for time? Good. I am gonna just spend 5 more minutes, then we've got time for Q&A. So if you like reading or watching, you can visit our website, sponsors.sanogenetics.com. There's a number of recommendations that I'll put on here. All these webinars that we run, we have recordings on the website that you can either read a summary or take a look at the recording. And there's guides and blogs as well. So take a look at our website. Also, there are a number of outlets I'd recommend. Things like Genome Web is a really good one if you'd like to get up to speed on some of the changes in the genomics and precision medicine landscape. If you like listening, I mentioned this earlier, but we run a podcast called the Genetics Podcast, so it's pretty easy to find. And there's 4 recent episodes that I think are going to be directly relevant to this group. So episode 94 with Paul, who's a leading target discovery at Alnylam. And we actually talked through this HA01 human knockout example. We ran a webinar and a podcast episode on genetic testing in ALS, comparing the US, Canada, and the UK, and what the similarities and differences are. And then a nice long 2022 recap of some of the most interesting discoveries with Vera. And then finally, Heidi is involved in one of the US-wide and really international programs on trying to determine which genes and variants ought to be included in commonly used diagnostic databases. So there's about 100 episodes that you can look at if you're interested in going into more detail in these kinds of things. So I'm going to stop there. We got plenty of time for a couple of questions if people have any. And thank you. I appreciate all of your time listening and for giving 45 minutes of your Monday afternoon or morning if you're in the US. So do we have any questions that anybody wants to put through the chat? Great. Mary Beth says, great podcast. Thank you. I appreciate that. Let me answer this one. It's important to, so this attendee says, although it's highly important to reduce the patient's diagnostic journey, I would find it important that the healthcare provider ordering the genetic testing can interpret all the results. How can we provide access patients to clinical geneticists quicker? Yeah, this is an excellent question. So in many cases, and I think actually in most cases, the healthcare provider may not be fully comfortable interpreting genetic tests or answering all the myriad of questions that have come. So there's a great paper that interviewed and sent a survey to around 100 Parkinson's disease professionals, so neurologists, and asked them about the challenges and barriers and what they struggle with or don't struggle with genetic testing. And the more than half of neurologists had significant challenges here. So I think it's a real problem. I think there's a couple things. First is robust support from genetic counseling. So both for patients and for healthcare providers, genetic counselors are highly trained in answering some of the common questions that come up around what genetic tests mean, what they don't mean, implications for family, and they can be a support to the healthcare providers who may be domain experts in the disease, but less so in genetics. So I think coupling healthcare provider support with genetic counseling support, whether it's available already at the healthcare location or providing it through a platform like ours or a pre-screening program. There is also a shortage of clinical geneticists and genetic counselors, period. So on a more macro level, I think it's a priority of most national scale genomics frameworks to increase the number of people that are working in this kind of field. But until then, I think there's also other areas technology can help, like providing access to educational information that helps to reduce the total amount of time that healthcare providers or patients need to cover the more basic questions, so that more time can be spent on some of the more challenging ones that really can't be covered through a webinar or a blog post. Great, thanks for that question. Daniel asks, how do you collaborate with population-wide genomics initiatives like Genomics England? How does Sano fit alongside them? Yeah, great. Thanks for that question, Daniel. So one of the things that we do working with population genomics initiatives is provide an interface for participants to answer questionnaires, to get recontacted about future research opportunities, and serve as that digital front door, for lack of a better word, for these population genomics initiatives. And this is an area we've run a couple of webinars on in the past, if anybody wants to take a look at how these work. It's a growing phenomenon in the industry that many countries, or in large countries like the US, there's both country and often state or health system level efforts to do genetic testing on a really large scale. And as I mentioned earlier, there's a unique aspect of genetics, which is that you really only need to do it once. There's an asterisk here because there's the technology is going to change and so on. But I see a future in the next 10 or 15 years where every person in many countries that have access to the technology and the means to do so will be offered genetic testing either from birth or from a certain age in adulthood, because it's, as you can see from the graph, it's clearly becoming cost effective. And if you only need to do it once, then it can serve as the backbone of a lot of decisions. So I just want to say this as a background for a broader trends for those of you all to look at. And these population genomics programs and healthcare systems are a key part of that. Great. So Mary Beth asks, can you speak to the movement to pay people for their DNA? Yeah, I think this is a great one. So in clinical research more broadly, there's ethical frameworks for when you can and can't pay participants to take part. So you can reimburse for time, you can reimburse for expenses, and this is typically something on a case-by-case basis that's handled by ethics boards. One of the big questions that's always been around in the field is how much should participants be entitled to reap the rewards of discoveries that are made using their DNA. My viewpoint on the issue is that I think there should be legal structures in place that allow companies that are doing work in genetics to share value back with participants who take part. I think it's a challenging set of incentives to align, and one of the challenges is also practical around how do you actually do this in a way that doesn't induce people to game the system and so on. But I think at a principle level, it would be really exciting to see. And there are some companies like Variant Bio and some others that are doing this on a small scale with some really interesting approaches. It's something I think a lot about. There's a lot of both legal and operational challenges around how you pay people, when you pay people. Is it simple, cash for your data, or is it a share in a company of some sort? I think it's a really fascinating area, but it's turning out to be, I think, a really tricky one for anybody to put a scalable solution in place. I think one of the more practical things that many biotech and pharma companies can do, and many are doing, is think about how they set up access programs for their treatments so that prices are realistic for patients who put their blood, sweat, and tears into taking part in the studies. And there's a whole, if you want to listen to our podcast, we've had a number of discussions about drug pricing and some of those other things. But I think a facet of this question is how do you make sure that patients who take part in clinical research are rewarded for and are able to have access to that drug and not priced out of the market when it comes to market. So I don't have time to do a full dive into this today, but I think it's an important area. Follow-up question to that is, speaking of returning value, how prevalent is the communication of the actual study results to participants? Yeah, I think this is another great question. What I think a lot of research programs actually fail to take this low-hanging fruit, which is a simple, regular communication of updates of the research that participants are taking part in. And oftentimes there are some challenges that make this difficult. If it's a blinded clinical research study, you may not be able to do this until the data is actually analyzed. But I think a lot of researchers could make a better effort to communicate the results of the studies more proactively to participants. And one of our roles as a company is creating the infrastructure to do this. So I'm talking my own book a little bit in this, but I do think it's important that actually a really small amount of effort can go a really long way as a participant, making you feel like you're you really are part of building something, and that you haven't just given a sample and you never hear anything again. Another question about saying, I do agree the cost effect may allow us to do general genetic testing, maybe even from birth, but there's still a lot of bias that sometimes keeps people from wanting to be tested. How do you think this should be approached so that they have full knowledge and are open to testing? Again, another excellent question. We know that as a whole, genetic data is not representative of the world we live in. It's much heavier tilted towards people of European ancestry. And there's a lot of reasons for this, including where studies have been run, but also historical research malpractice that has alienated some groups. So I think the industry has a lot of work to do, and we've written a couple things on and ideas on how to approach this actually from a funding level. But I think that it needs to be led from a community level, and we need to not assume that everybody has the same reasons for not taking part in testing, and think about solving the problem holistically. That how do you actually understand the problems that particular communities have that would stop them from engaging in genetic testing, and how do you help to improve the process and improve the service so that it's open to them. And some of the questions that come up that are extremely legitimate all the time is, who's going to know about this information? What impact is it going to have on my healthcare? This is very common in the US, of course. Also, there are questions that are universal about family members. So what's going to happen if it's a devastating result? How am I going to communicate it to my family? And again, building educational campaigns around these kind of programs that try to build that support net, I think, are an important thing. Great, thanks for that question. All right, great. Well, thanks, everybody. We appreciate your time, and thanks for signing up for this. Any feedback that you have would be great. Feel free to email me directly. You've got my email up there. And I hope everybody has a good Monday, and we'll send you a message the next time we have one of these webinars going that you're more than welcome to attend.